So this was the original title of my talk, but then yesterday, after seeing the first slide of Akshit, I realized that we were very much convergent on the same title. So the difference was that Akshit's title was Ecology and Evolution in Any Dimension. Mine is Microbial Ecology and Evolution in Any Dimension. And so I decided to change it, and thinking about actually what I'll show you is actually a more appropriate title is Microbial Evolution without dimension, because actually I think what we are trying to do in some sense is always to simplify or to reduce the dimensionality, and in some sense, at least for the first part of the talk, is like really remove dimensions from the game. Okay, so the starting point is, of course, the diversity and the complexity of microbial communities as a prototypical example of highly diverse communities. And what we know is that microbial communities are complex, their composition change over time, they fluctuate a lot. And in these fluctuations, of course, the natural things that you immediately realizing, which is very appropriate for this meeting, is that the reason why they fluctuate and the complexity that emerge from these fluctuations that gives rise to this complexity is actually that the environment they live in is very complex. So if you have a microbial community, whether it's in the gut, in the soil, in the ocean, and you try to characterize what is the environment, what are the factors that determine they grow, you realize that there are too many to track them. And even if you are in the lab and you try to do an experiment, you change a little things, you change the type of water you use, you add a little bit more salt, then you get like incredible effect that you were not expecting, right? So the environment is very complex, and it's very complex not just in the sense that there are many things, is that these things affect how bacteria grow. Now, these microbial communities are complex because there is not just one organism, there are many organisms, and these organisms are complex. So the trait space is very dimensional. This is what was also described in the previous talks, that there are many things that bacteria do and can do, and all these things contribute to how they grow. And sort of this, if you think about, is the triple helix that Akshit discussed yesterday by Lewontin on gene, organisms, and the environment. Okay? So this talk is divided in two parts. The first part is actually much shorter than the second one. And the first part is sort of try to either forget or embrace this high dimensionality and focus on what are the typical trajectories of these populations in empirical microbial communities. And the second part is more theoretical on evolution in this kind of uncertain environments. Okay, let's start for the first part. And the question is, what is the typical dynamics that we see in microbial communities? And one thing that I wanted to mention as a challenge in general in studying ecology of diverse, but also of non-diverse community, even of two species, is that in all the other, I think in all the other systems that we see here, whether we look at cell physiology, immunology, neuroscience, it's sort of natural to think that there is an underlying objective function. I mean, this objective function can be unknown, can be complicated, but it's clear that the things that are interacting in this high-dimensional space are performing one function. Or at least they are they're performing many functions, but at least they're contributing to the survival of the individuals and to their long-term fitness. Okay. Now, this function can be complicated, it's not simple. When you are dealing with ecological communities, there is not such a function, right? There is not a function that the community is performing. I mean, the community is performing functions, but it's not obvious that there is some underlying optimization mechanism. There might be conflicting optimization mechanism of the different members, but we cannot sort of consider this global objective, close parentheses. Anyway, so the question is, what is the typical dynamics of microbial communities? And sort of I'll tell you what is the simplest wrong model that describes the communities, and is a very simple model which basically describes the population abundance of one typical population in these communities. And there are basically two terms. One is very simple, logistic growth, these species have some typical value they fluctuate around. And the other term, which is the source of noise, is a fluctuating term with multiplicative noise, and this multiplicative noise takes into account fluctuations of the growth rate. And so the idea is the sort of, and I'll explain why I think this is the simplest useful wrong model to describe these communities, is that this model captures two essential ingredients that we see in these communities, that populations have a typical value of abundance they converge to. There is something in the environment that limits their growth. And this environment is fluctuating. It's fluctuating in this very high-dimensional space, and so it's affecting the growth rate of these populations. Okay, why this is a good model is because if you look, I mean, one reason is that there are sort of clear patterns that emerge in natural communities. So these are data of microbial communities from many different environments. And what you see is that if you focus on a typical species, the fluctuations that you see are very well described by a gamma distribution. And the same applies if you look at one population over time, for instance, here across host in the gut microbiome. And this is what you expect from the previous model. So basically, the previous model, as a prediction, predicts what you observe in these communities. And this model, being dynamics, also predicts some properties of the dynamics. So predicts what properties, moments of the transition probability. So if you take a species in your gut and you say, the species in this gut has this abundance today, what is the probability that this is going to be the abundance tomorrow? So in particular, you look at the first moments, you can also look at other properties. This model is in agreement with what you see in the data. Okay? And sort of you can build on this simple description and identify other statistical patterns in these communities. So basically, you can show that in some sense, in a nutshell, the parameters of these models that are not a time scale, so these carrying capacities and this variation, have also characteristic distribution across species. So there is a characteristic distribution of the variation of these across species, and there is a characteristic distribution of the variation of this noise across species. And sort of, yes? This one? CI, which is white noise. If you put some correlated, and the time scale of correlation is not too long, I mean, the stationary distribution does not depend much on that. The dynamics a little bit more, but. So the bottom line here is that, yes, these populations are sort of fluctuating and responding to fluctuations in this varidimensional space, but somehow if you forget about the dimension, what you find is that the underlying variation that you see in these populations has characteristic distributions. And these characteristic distribution in this picture are coming from some characteristics forces that are sort of basic ecological processes. There are limiting factors, so species don't grow indefinitely, and there is fluctuations of the environment. Yeah.
About what the data is here?
Okay, yes, I was going fast because I wanted to go to the second part. But yes, this data is data that you get from sequencing an environmental sample. These are, for instance, you can imagine in the gut, you have different individuals, and then you have different species, where species are like OTUs or amplicon sequence variants. You can do this in multiple ways, or even like at a finer resolution. And then you have the relative abundance, actually the number of reads corresponding to that. So you might see that a species is absent, sometimes is present, and is present with a different abundance.
Variance here is across different individuals.
Yes.
And there's a particular parameter tau I, ki, sigma I for each.
Yeah. So basically, the idea is that these fluctuations have a characteristic distribution, and this distribution depends on two moments: a mean and a variance. And these two moments are related to each other. Okay. So meaning that the coefficient of variation does scale with the mean, and the average as a typical distribution across species. And so you can translate this statistical observation of this variation into what is the, if you want, a prior on these parameters. Right. So basically you can say, suppose that I take a species at random from a community, what is the typical dynamics? Well, I generate at random a parameter ki from this distribution, this sigma from this distribution, and I run the dynamics, okay?
Thank you.
So it's a way to generate the ensemble of the trajectories. Okay. Now, why this is useful? Well, I mean, there is an interesting part that, of course, you see this sort of picture emerging. And the other interesting part is that you can use it as a lens to identify deviation. So I say that this is like the simplest wrong model, and the fact that it's wrong is what it makes it useful, right? And for instance, this is one thing that we did, was to use this model to discover what are deviations, and the most prominent deviation you see from the prediction of this model is like exemplified by these kind of trajectories, where you have a given population, and this population is fluctuate around a value, and then suddenly switches to another value. So you can imagine these as rapid switches of this parameter k, and you discover this by knowing that there is the model underlying, and so you can discover this deviation. And what we found is that these times where there are these transitions are like, of course, at specific times, they're not uniformly distributed, right? So there are groups of species that cluster together. And what is important to notice is that these are sort of unperturbed individuals, so they were not like subject to major infections, they were not taking antibiotics, they had not major travels. So this is like sort of showing that there are these switches, and actually one can show that a main component of the variation across the individuals is explained by these switches, that sort of two individuals are random realizations of these switches. Yes? Yes? What were the two? It's hundreds. It's a small fraction. Yeah, it's always, I mean, it's always less than half. Usually it's about 10%. But this is like over the course of 1 year. Yes, it's asymmetric. This is one example, but can go up or down. And okay. So this was, with this I wanted to show you some example where sort of you start with this very high dimensional structure of the composition, and despite or because of this high dimension, you can sort of discover these statistical patterns which are actually very robust. So this is, the first part was very short, and the conclusion is that basically these high dimensional ecological dynamics lead to these reproducible typical statistical patterns, and there are two basic ecological processes, sort of limitation from some limiting factors and environmental noise that capture these statistical properties. And somehow there is a hierarchy of mechanism, which is reminiscent of, I think, sloppy models, if you know what they are, that basically 90% of the variation is explained by these, and then the rest 10%, of the rest 10%, 9% is explained by a second mechanism, which in this case corresponds to alternative stable states in the human guts, or these switches of the typical value. Yes?
Can I ask a question? So given the data, is it possible to figure out whether noise is correlated among species?
Yes. Yes, it is. Yes.
Is it possible? And
yes. So another sort of things that we did was to look at the correlation across species, which is completely neglected to what I explained now. And the things we did is, I mean, there are correlations, so species are not independent. One thing that you can do is you can ask, are similar species more correlated or less correlated? What you would expect by chance? Yeah.
When there are these collective switches, so do these species have similar dynamics? Even
okay, very good question. We tried to understand this. I mean, one question that we tried to answer was, are species that typically have positive correlations or negative correlation when they are unperturbed, the ones that switch together? No. I mean, there are too few of these switches to detect this. Other questions? Good. So I'll go to the second part. And the second part is sort of focusing more on physiology and evolution, and is a very sort of a question that has been asked many times, which is how does a population adapt in uncertain environment? And this sort of, you have the question on the previous part? Yes. Yeah. No, there is no immigration. So yes, the point is that from what we see in the data, where something is zero, is very well explained by things that were not sampled. So if you want, you can imagine that you have a distribution of abundances, and this distribution is chopped because of sampling, or it's like convoluted with sampling, and you don't see that the part that is missing is different from what you have, right? So, okay. Other questions? Good. Okay, so this is work together with Romero, Onofrio, Mazzarese, and Martina Dalbello, and sort of it starts from an observation, actually, a set of experiments that shows that bacterial population can anticipate changes in the environment. So I want to show you a couple of these instances where this was shown. So this is one observation, that if you look at which genes are expressed in different conditions, you see that there are interestingly unexpected correlations. So for instance, if you look at the correlation of the genes expressed on a gradient of oxygen level, you see that that correlates with the genes that you see differently expressed on a gradient of temperature. So basically, the genes that are expressed when you decrease oxygens are correlated with the genes that you express, or an increased expression level of the genes when you increase temperature, which is this big bar, okay? So imagine that you have two conditions, and you high oxygen and low oxygen, and you look which genes you express, you have this variability, and then you look at what you do when you have low temperature, high temperature, and these expression levels are correlated, okay? So the question is why, and does it reflect something, right? Does it reflect some underlying non-trivial biochemical constraints that for some reason there are like constraints on what you express or constraints on the performance of enzymes on the presence of oxygen and temperature? And what they showed is that this is not the case, that you can, if you evolve a population in an environment where you have an opposite association between these two, so you have high oxygen in high temperature and low oxygen in low temperature, you decrease this correlation, okay? And so basically there is not a sort of biochemistry reason of why there is this correlation, is an evolved trait that you can lose. Yeah.
Just to understand maybe this is premature, but this sort of anticipation angle that you were giving, that these are two environments that are presented together, right? Rather than one.
No, in this case, so the first plot on the left.
Yeah.
There is like, no, I mean, I have a fixed level of, I have a fixed temperature and I change oxygen and I see what is changing. Then I have fixed oxygen and I change temperature and I see what is changing and they calculate the correlation, okay? So there is no reason why they should be.
Good,
yeah.
okay.
And so if you evolve in an environment where you typically have the opposite association, you lose this correlation, okay? And sort of what is the interpretation of this correlation that you observe? Well, you can think about the life cycle of E. coli. What E. coli likes to do is to enter into the mouth of animals and then go into the gut, right? And before entering the mouth is in an environment with typically a lower temperature than what is in the body of an animal. So the increase of temperature means that it enters the mouth, And what is going to happen next is that it's going to pass from an aerobic environment to the anaerobic environment of the gut. Okay? So in the typical environment where E. coli lives, which is not the lab, there is this association. Okay? There is an association between encountering higher temperature and a decreasing oxygen. Now, this is like based on a correlation where you see these two things that are correlated, right? And it's not obvious that there is a directionality. Now, there is another experiment which is very similar in spirit on sugars. So, E. coli in the gut sees lactose, typically lactose, and then maltose. And the observation is that, so each color here represents a different experiment run on different sugars and on different nutrients. And what you see is that when E. coli, and these are different genes. So these are genes that E. coli needs to grow on maltose and these on lactose. So when it grows on lactose, it expresses a lot of the genes on lactose. When it grows on maltose, a lot of these genes to grow on maltose. And now the genes that are expressed for maltose when grown on lactose are at a low level compared to what you express on another nutrient. But then when you grow on lactose, you express more of the genes for maltose than you would express in this baseline nutrient. And again, what you can do is that you can evolve E. coli in an environment where there is not this association, and if you do that, you lose the association. Okay? So the idea is that somehow E. coli, when growing on lactose, so in these red points, it expresses more, starts expressing something to grow on maltose even before it's exposed to it, in preparation for maltose. Okay? Okay, so how this anticipation is implemented, of course, there is an underlying complicated regulatory network which sort of implements these decisions. Okay? Now, the sort of simplified view in which I want to study this problem is a problem where a situation where there are different conditions, let's say two conditions, condition A, condition B, and our population of bacteria grows happily in a given condition, in condition A. Then there is a rapid switch of condition. And then what happens, this is also what is observed often in experiments, there is a time where bacteria sort of don't grow, or they grow very, very slowly in preparation of the new condition, and then they start growing exponentially. And so the two fundamental quantities here are a growth rate, how fast you duplicate in a given condition, and the lag time, how much time does it take to start grow when the condition change. Okay? No, not necessarily. Okay? Actually, I'll go back to your question in a few slides. Okay? Now, the simplest, actually this was a question for you. What is the simplest phase to study? But I already told you, the simplest phase to study is exponential growth, right? And there is a deep reason of why exponential growth is the easiest condition. And so the reason is that, suppose that you have an experiment and you have a colony that is growing, and you track how many copies of each molecular species you have in this growing colony, right? You will have some dynamics, which is complicated, is determined by this regulatory network that determines how many of these particular proteins you have in this growing colony, right? Now, if you assume something that sort of, that your culture is extensive, meaning that if you have something that is 10 milliliters and you cut it into the phenomenology you see in 5 milliliters is the same that you see in 10 milliliters or in the two parts, basically, this is the only form you can have, right? And now you can say, okay, where this phi is a concentration, right? And then you can say, what is the form of this concentration? And then you can derive this equation, and you get this equation here. And what you immediately recognize from this equation is that exponential growth and being in a steady state of concentrations are two equivalent conditions, okay? So it was interesting when Simone yesterday saying about when I studied statistical mechanics, the concept of equilibrium was always there, and then I study growth. In some sense, exponential growth is equilibrium, right? So it's exactly the same thing. Even if everything is dynamic, everything is growing. And this is captured very well by this famous quote by Leo Schachter, who say that the difference between exponential phase and balanced growth is the same difference between watching apple falls and thinking of gravity, okay? Which I think it's like give a sense of how central this is for physiology. Okay. And so in these simplified pictures, if we want to study evolution and we want to study evolution of anticipation, of course, where evolution acts on? Well, of course, you have like the genotype, and then this genotype affect what? Affect how these complex regulatory networks act and basically affect this function G, right? And so this is a complicated problem of understanding how you go from, of course, mutations in DNA to a changes of this function G, right? Now what you can say is, at the end, I mean, I can take the simple problem, which is to say that in exponential growth, right? If I'm in exponential growth, let's say the relevant phenotype that I need to understand how evolution is operating are these phi star, are the these steady states of this concentration. Okay? So this is about the simple phase, which is exponential growth. Now, I said that I wanted to consider two parts. One is exponential growth, and the other one is lag time. What is lag time in this picture? Why there is lag time? Of course, this is not an exponential phase, so it's not at steady state, which means that the internal concentrations are changing, right? And how they are changing? Well, the idea is that in condition B, you need some enzyme and transporters for, let's say, nutrient B, or if this is like not a different nutrient, but it's a different condition, you need to express something to deal with this different condition. And at the beginning, you have a low concentration of these, and then this concentration is increasing, right? It's increasing to arrive to the value that allows you to grow fast enough, to the stationary value in exponential growth, okay? And so how do we describe these dynamics? Again, this dynamics is complicated, yes. So my idea here is that the population is always homogeneous, okay? And so you have, I mean, then it's gonna evolve, but let's say for now, let's think that the population is homogeneous. They have all the same, it's a clonal population, and they do exactly, they are like also phenotypically identical. They do exactly the same, right? Which, of course, can be broken, and it's very interesting to break this. Okay, so now what I have to describe is what is the dynamics of these phi. Right? And in principle, this depends on the regulatory network. The idea here is that what is the limiting process that, I mean, what the cell has to do here, or a population, I mean, cells within a population have to do, is to increase the amount of these enzymes, right? And what is limiting the amount of these enzymes is the fact that you change the conditions, so are the enzymes themselves, right? So the idea is that there is some dynamics that I can write like this, where the dynamics of this phi depends on phi itself, okay? And sort of is growing and is up to the point where it is reaching the value that is achieved in balanced exponential growth, okay? Now, what you can do is that you can derive from this simple equation a lag time. And in particular, the lag time depends on the initial condition. So what is limiting here is what is the initial condition, right? The smaller is these things initially, the more you have to produce, and the longer it takes to achieve exponential growth. Okay. But where this initial condition comes from, well, in my framework, the initial condition is the condition that was in the previous condition, okay? So the initial allocation where you have the switch comes from what you have in this condition. Right? And the reason why you have that is because here I'm talking about rapid switches that couldn't be anticipated. I mean, that you are trying to anticipate, that bacteria try to anticipate, but they are sudden. There is nothing in the environment that is slowly changing, telling you condition B will happen. Okay? Good. So basically what we have here is that the larger is this concentration here when condition B, where the purple condition arrives, the lower blue had to be when blue condition was present. This, of course, where I'm slowly going towards to, is that there is a trade-off here, right? And, and the trade-offs come from the fact that basically what you can imagine, and this is sort of building on the framework that Sid explained, is that somehow you have to choose that the growth rate in the previous condition depends on how much of the blue enzymes you have, right? And if you increase the amount of purple enzymes, you have to decrease the blue enzymes, okay? So if you put these two things together, you can imagine that you have like a continuum of alternative strategies, but you can, if you are in condition A, you can allocate a lot for the present condition, having a faster growth, and the price you pay to have an egg today is that you will not have the chicken tomorrow, meaning that you have a longer lag, okay? So this gives rise to this trade-off where basically, if you track how much you allocate for the presence versus how much you allocate for the presence plus the future, yes. This is like buzzwords in the world of resource allocation models, is you have to produce, a growing cell produce express proteins, right? And what you track is what fraction of proteins you produce. I mean, you always produce proteins, and since you are in this exponential growth, the only things that matters are relative concentration, okay? So relatively, if you produce a little bit more of A, you have to produce a little bit more, the less, relatively, by definition, okay? So this balance tells you that you can, since you're looking at mass fractions, at concentration, and the total concentration of all the proteins is determined by density, is fixed. If you have more of A, you need to have less than the rest, okay? It's because is it clear or, okay. Good, so basically the picture here is that you can track what is the fraction that you allocate to the present versus the future, and if you allocate more for the present, you're gonna go faster in the present, but then you're gonna lag longer in the future, and what it comes out is a relation between how rapidly you adapt to the new condition and the growth rate, okay? Good. So another way to look at this, one way to look at this, and this is like related to the question about different nutrients, is that you can look at different population, which is what I'm gonna do in the rest of the talk, but you can also think about whether different conditions, so whether the egg was a quail egg, an ostrich egg, or a chicken egg, and the condition might change, right? And this is exactly something that they did in this experiment. And so one thing that is very well known is that E. coli grows different in different nutrients, and in particular, it grows quite fast on glucose and quite slowly on mannose, okay? So the growth rate is twice as big in glucose. The replication time is half, right? Then sort of the standard picture of why that difference occurs, which emerged like from the picture that also Sid explained, is that basically you have this different nutrient quality. So if you track how much you allocate, or if I use how many enzymes you produce to grow on per enzyme, the amount of growth you produce in mannose and in glucose, it's very different, okay? It's much more efficient to grow on glucose than on mannose. This is the sort of standard picture. And so basically the growth rate in mannose is lower because it's a less efficient nutrient. And what they did in the group of Marcos Bazan was to construct a strain that express the enzymes and the transporter for mannose in the same way as it expressed the transport for glucose. So basically, what you do is that you express the enzymes and the transporter for mannose in the same way you express for glucose. Okay, so you rewire the gene regulatory network such that the errors that goes into the expression of glucose goes into the expression of mannose. And what you see is that this mutant grows on mannose exactly, I mean, at the same growth rate as it grows on glucose. So basically, the difference between mannose and glucose is not driven by this different nutrient quality, but is driven by allocation, by the fact that you decide to allocate less in mannose than in glucose. And what is the reason and the consequence of this is that when you grow in mannose, here the orange circle, you have a much shorter lag when you switch, for instance, to acetate than you have in glucose, right? And now if you play this game here, you see that a longer lag emerged, right? So basically you have this effect of allocation. Okay, good. So the question that I want to ask in the rest of my talk is, based on this picture, how does allocation evolve in uncertain environments? And so the typical dynamics that I'm going to consider is these boom and bust environments where bacteria are added to some fresh medium with some amount of nutrients, and then they grow, and then they are a small fraction of them, 1/d is taken and put to a new nutrient condition with fresh nutrients, and then they grow, and so on and so forth. Okay? And so the first case I want to consider is a boom and bust environment where you have two alternating conditions, blue and purple. Okay? So blue and purple always follows blue, and blue always follows purple. So everything is deterministic. And so what evolves? Well, of course, there is a big engine underneath, but for me, what is going to evolve is this fraction f. Okay? And so you can ask, if I play evolution in this scenario, where I have this population growing, and then I add a mutant with a different value of f, and then this mutant will be subject to, well, selection, and I'll track the dynamics, what do I see? Well, the first observation is that the average value of this f in a population always converges for a fixed set of conditions to the same value, irrespective of where the population started from. Okay? And what does this value where it converges to? And actually, the population is homogeneous, so the variance within the population is very low. Okay? And so, what determines this value of f? Well, it depends on the parameters of my models, which are the dilution factor. So, how big are these bottlenecks? So, if I increase the dilution factor, I typically want to allocate more for the present than the future. The lag time scale, which sets what is the typical time scale of lag. If lag is longer, I want to allocate more to the future than to the present. And how fast is growth if I was allocating everything to the present. And so this dependency can be captured by a very simple parameter, which is a ratio of 2 time scale, which is basically the ratio of the time scale of lag and the time scale of growth. Okay? So how much time does a cell spend in the growth condition, and how much time does a cell spend in the lag condition depends on whether you want to allocate more for growth or more for lag. The more you spend in the growth condition, the more you want to allocate for growth, so for the present versus the future. And now, what is interesting here is that and depending where you come from, this can be very surprising or completely obvious. These populations do not maximize their growth rate. Right? So during evolution, what you see is that the populations that emerge are not the ones that maximize growth rate. So what is happening here, where you can use the framework of adaptive dynamics to understand what's happening. And actually, what you discover is that what happening is very simple. And what these populations are doing is that actually what matters here is not the growth rate, but is the time that it takes to deplete resources. So if you look within a cycle, there is a time that it takes to deplete resources, which is the lag time plus how much time it takes to deplete resources when you're growing. And you can show that strain will always invade with a given F, will always invade a resident population if this strain has a lower depletion time. So evolution is not maximizing growth, it's minimizing depletion time. And so basically, if you minimize this function, what you get is exactly what I showed here. And evolution is sort of climbing, or sorry, not climbing, descending this gradient of depletion time. Yes. Mm-hmm. Yes. Yes, that in some sense is always true, right? The point is, it's always true that you maximize your long-term fitness. The question is, what is the long-term fitness in terms of what? Yes, yes. Good. Yes. Because that's a very extremely good question. I mean, we are playing with that. The reason is that I'm considering an homogeneous population. So you can actually ask, is bet hedging advantageous? And the answer depends. And that would require that the distribution of this F is not a delta function. So here I'm considering that there are mutations, right? And mutations give rise to moving this delta function of F, right? So if you have a given genotype, you have a fixed value of F. Right? Then you can ask a second question, which is it advantageous to have a distribution of F? And the answer, whether it's advantageous or not, depends. Actually, in this specific setting, is not advantageous. Other questions? Okay. Now, what you can say, okay, this is like interesting, but it's a deterministic environment. What happens if I have a stochastic environment? And in this case, stochastic means that there are many types of nutrients, right? So blue can be followed by purple, by orange, by green, or by yellow. And when I add new resources, the amount of resources is not fixed, can vary. And when I dilute, I don't take a fixed amount of cells, but I take a variable fraction, right? And I can ask what happens there. And so what you see is that nothing changes. So basically, if in the deterministic case, the condition for invasion was that the invader had to have a lower depletion time, now long-term fitness is maximized if your expected over the stochasticity depletion time is lower than the one of the resident. Okay? So in this, if this expectation is lower, then you invade. Now in this case, this is where bet hedging could play a role because of course, in that case you have a distribution and that matters, okay? And actually that noise can be beneficial. Questions? Yes. No, in this case, any condition can follow from any other else, but I'm going in that direction, so let me continue. And so now in this case, basically what you have is that you have the same picture as before. Now you have M conditions and you can switch randomly between them with no structure. And in this case, you get exactly the same as before, right? Where basically the only difference is that here you have the expectation over this log dilution. Okay, good. But now, in this case, I have assumed that every condition can follow from any other else. So orange or green have the same probability of following blue, right? And sort of what I've studied so far is how much you allocate for now versus what you allocate for the future. But of course, here you have many potential futures, right? So if you now zoom in into what you allocate for this potential future, you have many options, right? You can allocate for orange, for purple, for blue, for red, etc. And so, and the point is, you can imagine that there is some statistical structure, right? So the probability that purple will follow blue is not the same that green will follow blue. So you can encode this in some transition metrics, telling you what is the probability that if I see blue, purple is going to happen. Okay? And there are different degrees of predictability. So you can go from a full cycle where everything is perfectly predictable to like everything can follow anything else where everything is unpredictable. Okay? And similarly, if you look within this sector, so now you take this sector that is allocated for the future and you look at fraction, and it's interesting that this game is looking at fraction, then fraction of fractions, fraction of fraction of fractions, and I think that this is very important, but you look at the fraction within these things that you allocate for the future that you allocate for any of the potential futures, right? You can allocate for purple, for yellow, or for others. And of course, if you are in a different conditions, you will have different allocation strategies, okay? So what you can ask is playing the same game that I played before, right? You have this evolving population that now can mutate, well, the value of f, but also can mutate this value of q, how much you allocate for any of these potential futures. Okay? And so you start initially that you are in an environment, and of course, you chose a random initial conditions, or the two things are independent, and then you evolve, and what you get is that you sort of match the environment. Okay? And the reason why you get this is because, you can understand what is happening by looking at the population average of this allocation and studying their evolutionary dynamics using adaptive dynamics. So basically in adaptive dynamics, what you do is that you ask, will an invader that is very similar to the resident population will invade or not? And you sort of climb this local gradient on whether of invasion fitness, of whether you're going to grow if you have a slightly different rate than the resident population. And in this setting, what you discover, if you write this equation, is something that is very nice to write in some sense, that these allocations, the average trait, climbs the Kullback-Leibler divergence between the statistics of the environments and the allocation. Okay?
I have a question.
Yeah.
In all of these models, one would have to assume how much cost you pay in expressing genes associated with that.
So this all depends on
the H function.
Yes, on the H function, exactly. On this H function.
And in these models, like what would you assume?
So in the model I showed you, this H function is, it's very clear why the Kullback-Leibler emerges, because I assume that H is linear. And so the lag time is logarithmic, and that's what gives the log that appears in the end. Now, if you assume that this H is a power law, this, let's say the linear case is the simplest to imagine, in the sense that you say, I'm sort of having a flux of let's say, mass and energy that I get from the environment, which is proportional to how many enzymes I have. There are cases where that is not the case. For instance, if you go from a glycolytic to a gluconeogenic resource, what you have is that this is phi squared.
So for the data that you showed from Basan et al.
That's phi squared.
That's
yes.
phi squared. Okay.
But honestly, in that regime, you don't see really much of a curvature. Yeah. There is a scatter of the data. Yes, that's true.
Thank you.
But then if you have phi squared, what you get is that you are not climbing the Kullback-Leibler divergence. You have like a generalization of these Rayleigh entropies, and basically you are climbing a gradient of the cross Rayleigh entropy. Okay. Yes. Okay. Good. And so basically you match the statistics of the environment, okay? And now, as I say, this is shown for this linear function. In general, you can use different alternative forms. And basically, you are always minimizing some form of information between this Q and the statistics of the environment, okay? And also, this is reminiscent of what is called the Kelly strategy in betting, which basically say that under uncertain environments, you allocate proportional to the likelihood that a given outcome will occur. And the reason you don't want to allocate, for instance, for the most likely outcome in bet hedging is that, sorry, not in bet hedging, in Kelly betting is that if you allocate to only one, you're going to lose everything. And the same is here. If you don't allocate something for one of the potential future, will not be able to grow, and you'll spend infinite time lagging. Okay, so it's the same. It's similar in spirit. Okay. So now you can also like extend this a little bit to say now, let's say that these different conditions, you typically add different amount of nutrients, or you dilute at different values. What you have is that a small modification where now not only the likelihood matters, but always how fast you grow matters. And basically what you discover is something very simple, which is that the allocation are proportional to what is the expected future times the growth. So you want to allocate more for more likely and faster growing conditions. Okay? Yes. That's unlike Kelly, because in the, yes, that is where, yes, in that case it's different. Yes, because no, but I mean in Kelly it's not simple where you have multiple. So in Kelly actually applies for two outcomes where you lose everything in one, and when you extend it, it's a little bit different. Yeah. Yes. Okay. Yes. To Kelly? No, not exactly. No. I mean, let's say it's similar in the sense that you have something that very bad happens if you are at the extremes, if you go for the most likely one. There is a similarity, which is you have a log in between, and that is why you get the same case in the previous case. But then, if you slightly deviate, you have differences. Okay, so basically this is, I conclude, and sort of the conclusion of this part is that allocation to present versus future conditions determine this balance between growth in the present and lag in the future, and there is this condition-dependent optimal balance, and in this framework, I mean, this is like a natural framework to think about learning over evolutionary times and how populations can encode the statistics in the environment in their physiology. Okay, that somehow these pre-expression levels encodes what is the statistics of the environment they evolved in. Okay, so with that I conclude by thanking you, thanking my collaborator, and with a very short advertisement that we have. This is part of a project that, and we have an open postdoc position to work on these themes of the second part. So thank you very much.
All right, we have time for a few questions. Yeah.
Mm-hmm. Yes.
Do you mind repeating the question, Jacopo?
Yes, the question was why exponential growth and physiological steady states are equivalent, right? So the idea is, suppose that you have a growing colony, right, in a flask, let's say in liquid culture, and everything is well mixed, right? And you look at any given time how much of a given protein I have, right? This is growing, okay? And then you say, this is growing according to some rule, okay? Now, what I'm saying is, if I look at these and I ask questions, the answer I will get is independent of whether I'm looking at the 10 milliliters cultures I'm considering, or I'm cutting this in half and then looking at the 5. Right? This is like some extensivity conditions, right? That basically you look at the interface. I mean, it's the typical things to do with statistical mechanics, right? You have a gas, you divide it in two, and you say the property of the gas in one half are if it is are the same as the other. Right? And the point is, if you then write the dynamics of this phi, right? Just by starting by this definition and this definition, this is what you have. Right? Basically, you have that. So you have just to use the chain rule. You have dt of ni over v, and this is equal to ni dot over v minus ni over v dv dt. Squared. Thank you. Okay, and this is exactly what I wrote there. And so the point is, the fact that this is constant, right? So the fact that you have stationary concentration, if this lambda, so this derivative here, is not dependent on time. Right? So being in exponential growth requires that these two things are constant. Right? Yeah. Okay. So that is basically the condition. Yes. Yes. No, lambda is the growth rate, right? So if the lambda is dependent of time, yes. So basically what I'm saying is dv dt is equal to lambda v. So the condition for exponential growth is that these things is a constant independent of time. If this does not depend on time, you will see, if I was looking at v log v versus time, I will not see a straight line, I will see something else. Okay. Okay, so if you look at single cells, you can say, here I'm looking at bulk population. So I'm looking at the population as a whole. Now you can say, if now I want to go from single cell to the population, in some sense the same applies, because if you have individual cells, and if you say that if the individual cells are in steady condition, I mean, within a life cycle there could be things that changes, but if you have these changes are sort of steady, they don't change over the course. If the physiology is stationary, then you have exponential growth. Yeah. Yes, you can have. No, but it's right, but that is okay. Okay, good. But then I can think of that. Okay, that is in some sense is something that I did not hear, because here I'm considering an homogeneous population, right? If you have a population with different ages, you can imagine that is non-homogeneous. Here it's a homogeneous population, right? I'm saying I have an homogeneous population, I'm looking at the bulk, and I'm saying what the bulk is doing. Now, of course, if you have an heterogeneous populations, you have heterogeneity, and you are right in the sense that if you have heterogeneity, you have exponential growth of the total one, still having a variation at the individual level. But what you need to have this exponential growth being exponential is that this variation needs not to change over time. Because if the distribution changes over time, then you will see changes in the growth rate. You're not convinced, but then we can chat later. Yeah.
Okay, yeah,
Yes,
maybe,
but,
yeah, maybe one last question. Yeah.
Yes. Yes. Exactly. Yes, exactly. Yes, but that's true. So if you look at the total number, yes, but that is, I was thinking of that. If you have the total number of cells, you need to have that the condition that basically the total number of cells and the total volume changes with the same rate, right? Yes.
The volume of an individual cell.
Yes.
Then you have exponential growth.
Okay. Okay.
And concentration.
Yeah, okay, okay.
Okay, so if there are no more questions, we're out of time. You can always find Jacopo and Michael later. We'll break for lunch now. We'll come back here at 5 minutes to 2:30, 2:25, yeah.